Look at how many people ran over to speak to one of the leading analytical biochemists in this space. It is a nice reminder once again that marketers need simplified messaging, but we don't need oversimplified messaging. So far we're doing wonderful. Dr. Makarianis, what a great talk. Our next speaker is Dr. Josh Hartzell, and Dr. Josh is a Cannabis employee. He is with Cannabis Laboratories. And what I'm about to share with you before Dr. Josh takes the stage is something that took me a little while to get used to. I just recently resigned from Nordic Naturals. I worked in the research and education department for the last 10 years, and I spent my life obsessed with omega-3 biochemistry and science. And my job was to prevent people that work in health food stores from oversimplifying the science, which they do all the time. So when I was invited to join the Cannabis team, many of my friends and colleagues said to me, "What are you doing? This is the craziest thing you could possibly do. You have this absolutely secure position. Omega-3s, they're now 3 pharmaceutical drugs. There's 20,000 papers, 2,500 randomized control trials, and we pretty much understand how the stuff may work." And I told all my friends and colleagues, I'm going to go work for this hemp company that makes CBD. You got to try it. And they thought I was mad. Well, when I got to the laboratory, when I showed up at the corporate offices in San Diego and I got a chance to meet Dr. Josh, at that time, he had just received his 4th Cannabis Cup. Now there have been 5 High Times Cannabis Cups for the best CBD in the world. You know how hard that was for me to say in the beginning? I actually was a bit embarrassed when I said that, except yesterday was a nice reminder to me. So I don't think there's any shame to what we're doing here at all. This is amazing, amazing medicine that will help ease the suffering of millions and millions of people. So let's welcome Dr. Josh to the stage with his 5-time Cannabis Cup CBD.
Welcome. Thanks for coming. It's truly an honor to present here with some of my own heroes. And today I'm going to explore some of the phytocannabinoids and how the plant makes these compounds and some of the diversity within the plant. And I'm the Director of Research and Development at Cannabis, as Stu said. So here we'll start this off. Within the Cannabisaceae family, the genus of the Cannabisaceae, there's actually 3 different types of plants. There's Cannabis sativa, which hemp is part of that genus. Here we have a picture of me standing in a hemp field at Murray State, where we're growing one of the first agricultural hemp crops. We also have hops. Hops, many of you are familiar with because they're used in brewing beer. And they're actually so similar to one another that you can graft hops and cannabis onto one another. So they produce a lot of the same terpene type compounds, but hops does not produce cannabinoids. And recent genetic evidence has shown that this Celtis or hackberry tree is part of the Cannabisaceae family as well. So Carl Linnaeus is the father of modern taxonomy, and he has classified all varieties of cannabis as Cannabis sativa. And the L at the end of the name stands for Linnaeus, Carl Linnaeus. There's a debate about this. There's 2 different viewpoints on it. There's a monotypic view and a polytypic view where cannabis is either all one species, like Linnaeus said, and that's what we'll use for this presentation. Or some people believe that there's actually different species of cannabis, Cannabis indica, Cannabis sativa, and Cannabis ruderalis. But we'll stick with Carl Linnaeus's nomenclature and call everything Cannabis sativa. But we will talk about the differences between these subspecies. And Cannabis sativa is characteristic of these tall-like plants with long internodal distances, narrow leaves, and these plants originate from the equatorial regions of the world. Some of the examples of this are Acapulco gold and Malawi gold, and most of the hemp varieties are actually sativa. And the reason for that is because they're produced for fiber, and the longer the internodal distances, the stronger the fibers. We also have Cannabis sativa indica, and these plants originate from Asian regions of the world specifically India, Pakistan, Afghanistan, and they're renowned for their high resin content, high THC content, and actually Carl Linnaeus was not aware of these plants when he named cannabis sativa, so it may have been that he did define them as a new species. And some of these plants include G13, Hindu Kush, and blackberry, and they're characteristic for their short and broad leaves. And then Cannabis sativa ruderalis originates from Russia, and one of the interesting properties of this plant is it will auto-flower, meaning that most cannabis plants require more than 12 hours of darkness to induce flowering, but these ruderalis strains will flower on their own and are not induced by light. Most of the cannabis strains that you see in the industry are actually hybrids to express the desired traits of these plants. So what is the legal definition of hemp? I know Chris touched on this recently, but it's 0.3% THC or less in the dried plant material, with the exception of Italy, which defines it as 0.2% THC. And the United Nations Office of Drug and Crime has divided cannabis into 3 separate chemotypes. And the equation they use to define these chemotypes is the THC value plus the CBN value divided by the CBD value. And drug type cannabis is a chemotype 1, where the X value is greater than 1. Chemotype 2 varieties of cannabis are less common, and that is when the CBD and THC values are nearly equivalent. And then we have the fiber type hemp crops, where Chi is less than 1, and that is when the CBD values are greater than the THC. So currently there are 46 types of hemp that are approved by the European Union. With the recent farm bill that was enacted, now we can grow hemp here, but one of the problems is we haven't grown it in the United States so long, we don't know what varieties grow well. So that's what we're doing in Kentucky right now. We're really trying to explore that and find the varieties that will grow well in America's climate. Because in Europe, it's a higher latitude and it tends to be a little colder. So an interesting characteristic of the plant is the THC to CBD values are constant throughout the plant life cycle. So you can actually test a seedling and find out what chemotype it is. You don't have to wait till the plant is finished flowering in order to find out what type of plant it is. So agricultural hemp has a lot of interesting properties. It has 30 to 40% fiber that's used in rope, textiles, and building materials. And we see down here we have hemp concrete, hemp rope, and one of the interesting facts that I like to tell people is that Betsy Ross sewed the American flag on hemp. So we also have plastics for cars. We have hemp seed oil. And one of the interesting characteristics of hemp that we exploit is the high CBD values and very low THC values. And many of the hemp species, as I mentioned, are cannabis sativa, which dispels a common misconception that I hear in the industry. Indicas have high CBD values. Most hemp varieties are actually sativa, and they're the ones with the high CBD values. So I like to point that out to people. As far as constituents of the hemp seed, they're actually 20 to 35% oil, and they have about a 2-to-1 ratio of omega-6 to omega-3. They also have a lot of saturated fatty acids like palmitic, stearic, and oleic acid, but they also contain 20 to 25% protein, 20 to 30% carbohydrates, 10 to 15% fiber, and a range of vitamins and minerals that mainly include vitamin E, B vitamins, phosphorus, potassium, and a host of other elements. So what about the seed? Does it contain cannabinoids? They do contain cannabinoids, but they're not actually produced by the seed. They're actually a contamination product because seeds are produced in the flower and some of the trichomes fall off and they contaminate the seed. So here we see fiber type hemp is between 0 and 12 parts per million, whereas the drug types are a little higher because they have a little bit more contamination product on them. And then within the kernel, you see less than half a part per million. So the take home here is that cannabinoids aren't produced in the seed, and they're actually on the surface and can easily be washed off by an organic solvent. So Alex has already touched on this a little bit, so I won't beat this dead horse, but cannabis has over 525 compounds that have been identified to date. 140 of those are terpenes, and to date there's 86 cannabinoids that have been identified in the cannabis plant. There's 50 hydrocarbons, flavonoids, there's 23. Actually, these are glycosidic flavonoids. There's no free flavonoids that have been identified in cannabis. And a series of many simple compounds, small molecules like ketones, esters, lactones, and some vitamins that we've already discussed. So let's talk a little bit about the plant biology. There are two different types of cannabis. There's monoecious and dioecious cannabis. A lot of hemp varieties are actually monoecious, meaning that they produce the female and the male flowering parts on the same plant. And these are called hermaphrodites. Hermaphrodites are when you have male and female in the same plant. And on the female plant, you actually produce these pistils that can be pollinated by a male flower. There's pollen grains that come out. And this is where the seeds produce in the calyx. Now, many cannabis growers don't want seeds because if you produce seeds, then you take away from the resin content. The cannabis plant actually uses that energy and produces seeds instead of resin. So you want to eliminate males in a lot of flowering plants to not produce seed. And this is actually a sinsemilla flower here where you can see it's coated in crystals. These are actually trichomes that I'll discuss in a second. So trichomes are the cannabinoid and trichome factory of the plant. This is the most commonly discussed trichome. There's actually 5 trichomes in cannabis. This is a glandular trichome and it's called a capitate stalk. It looks somewhat like a mushroom. It has a stalk with a head, and these are produced extracellularly. And the reason they're produced extracellularly is because many people don't know this, but the cannabinoids are actually toxic to the plant. So they're produced in this extracellular space that's very oily. They're excreted up through this neck, and the plastids are where the terpene precursors are found. The vacuoles are where the olivetolic acid, the cannabinoid precursors, are found. They're excreted into this extracellular matrix, and they're combined where the enzyme, this is where the enzymes are active. The enzymes are excreted here as well, and the molecules are combined and synthesized within this oily extracellular space. But there's actually 3 types of glandular trichomes. The capitate stalks that we just discussed are only produced during the flowering stage of the plant. But there's also capitate sessile trichomes that are smaller. They produce cannabinoids as well, and they produce them throughout the whole cannabis life cycle. They're found on the stems. They're also found on the leaves. And we also have bulbous trichomes that are 2 to 4 celled structures that do not produce cannabinoids. And over here on the right, we have a microscope image of a capitate stalked trichome that develops during flowering. This is also on the leaves. And a scanning electron microscope, which shows the structure up close. In addition to these glandular trichomes, we have 2 types of non-glandular trichomes. These are unicellular structures. They look somewhat like hairs. And what differentiates the two is the cystolithic trichomes is a botanical term for an inorganic precipitate that's formed. And in cannabis, that inorganic precipitate is calcium carbonate, and it is at the base of the hair. And in the non-cystolithic trichomes, they do not have this, but they are a little bit longer than the cystolithic trichomes. And they're also coated in silica. One of the interesting things about the non-glandular trichomes is they're highly silicified. People believe that the trichomes serve multiple functions. And they're not just found on cannabis. They're found on lots of plants, algae, and protists. They disrupt the feeding behavior of herbivores that would want to eat them. They prevent frost prevention in later stages of the growing season. They provide a light reflectance property in desert environments. They prevent transpiration from the plant in desert environments as well. And one of the most important features, I think, of the trichomes is they emit compounds that deter insects, like terpenes, and they promote pollination. So we'll touch a little bit on these cannabinoid classification, and many of you probably know of THC, CBD, and CBN. But what you may not know is that there are many types of THC. There's many types of CBD. There's many types of CBN. There's actually 9 known THC analogs. And as Alex mentioned before, some of the differences here are the side chain. There's a carboxylic acid here and some of them that I'll discuss in a minute. But these account for some of the differences in the structure of the cannabinoids. In addition to these well-known cannabinoids, we also have other molecules like CBG, CBC, CBE, CBT, and even CBND, which has been debated in the literature due to analytical techniques at the time. No one knows if this really exists. And I certainly don't have time to cover all 86 cannabinoids. And there's also 29 other miscellaneous cannabinoids as well. So there are a lot of cannabinoids that can be found in the plant. Not all of these are found at high levels, of course. Some of these are found at very low levels in the plant. but there are lots of compounds that have been identified in cannabis. One of the interesting facts that I learned when I was at the ICRS conference with Alex is that cannabis is not the only plant that produces cannabinoids. Hylichrysum is a South African plant that produces CBG and CBGA. It doesn't produce any of the other cannabinoids, but it does produce these two. And it's been used in traditional medicine for a host of inflammatory, anti-inflammatory, and anti-infective conditions. And liverworts is another plant species that has cannabinoid-like compounds in them. Now, the plant doesn't actually produce CBD. It doesn't produce THC. It produces the acidic analogs of them, cannabinoic acids. And here we see in red this carboxylic acid group on THC, and we call this THCA. Notice when you heat it to over 105 degrees Celsius, the CO2 molecule is lost, and you generate a neutral THC molecule. You lose mass in the process. That's because of the loss of the CO2. This is also true for the CBGA, CBDA, and all of the other cannabinoids. So this accounts for the 9 known, when I say 9 known THC analogs, there's acids, there's non-acids, there's different linked side chains. And this is where we get the diversity within the cannabinoid family. Here we show again this 5-carbon side chain where we have a 3-propyl side chain, and we generally abbreviate these compounds as called varin. So when we have this 3-carbon side chain, we call it tetrahydrocannabivarin, or if it's CBD, it's called cannabidivarin. So this is probably the most common side chain in addition to the 5-carbon side chain, but there have been a lot of them identified. There's been 1-carbon side chains, 2, 3, 4, and 5 found in the plant. How are these compounds made? There's a series of parallel enzymatic cascade that the plant has going on at once. And like all plants, we take CO2 and water and sunlight. Photosynthesis makes sugar. The sugar undergoes a glycolysis reaction to form phosphoenol pyruvate. This precursor can be diverted through a number of different cascades, but one of them is to go to shikimic acid. This is a precursor to flavonoids, lignans, coumarins. This phosphoenol pyruvate can also be converted into acetyl coenzyme A. Acetyl coenzyme A is a precursor to polyketides and lipids, and it just so happens that one of those polyketides is olivetolic acid. And olivetolic acid is the precursor to CBGA, and this CBGA molecule is the mother cannabinoid. All cannabinoids are synthesized from CBGA. In addition to that, acetyl coenzyme A can be converted into geranyl diphosphate. Geranyl diphosphate can be diverted through parallel pathways. One of them is to make monoterpenes. Limonene synthase is an example of that. It can also be diverted into sesquiterpenes through FPP synthase. So there's only 3 enzymatic cascades that the plant uses to make cannabinoids. CBGA is either converted to CBDA, THCA, or CBCA. And here we show a crystal structure, or just a crystal, we show the crystals of THCA synthase. After that, it's a degradation process. So these 3 cannabinoids are made primarily, and the other cannabinoids are actually degradation products of these 3. So CBDA can be converted into CBEA through photooxidation or pyrolysis. THCA can be oxidized into CBNA, and then it can be decarboxylated if it needs to be. Heat can convert it into the delta 8 isomer, which is the thermodynamically stable product. And then CBCA can be converted through light into CBLA. So I want to talk a little bit about the different properties between THC and CBD because they have exactly the same molecular weight, 314.5. They have a nearly identical structure, but they have different pharmacological effects. Why is that? So right here we see in THC, there's a conformationally constrained rigid bond due to this ether linkage. So this molecule is stuck in this shape in 3-dimensional space, even though it looks the same on paper. In 3-dimensional space, it's very different. Because in CBD, we have a freely rotatable bond around this blue bond and 2 free hydroxyl groups. So this part of the molecule can actually flip around. It's kind of just wiggling out there. And we show this on the CBD low energy conformer, where this flips around. And this has important binding implications, because if it's different in 3-dimensional space, it's going to bind to the receptor sites differently. So some of the THC potency trends, if we look at them, have been going up over the years. Sinsemilla is shown here without males in the garden. This is all cannabis collected. You can see from 1980 to 2008, it's gone from 1.5% to 8.8%, with the exception of Sinsemilla. If we look at it, it's a little bit higher. And then we show domestic. This is domestic crops that have been taken, and this is foreign. So you can see they're doing a little bit better job than us as far as THC goes there. And CBD is a little bit different. The CBD levels have remained relatively low and constant. They're staying below 0.5% in all of the cannabis collected. And the same is true for CBN and CBC. So you have these growers out there that have unknowingly bred these cannabinoids out of the plant in lieu of high THC content. Now I'll talk a little bit about the terpenes. Terpenes are found at low levels in cannabis, less than 0.3% of the fresh weight. And most of these terpenes are monoterpenes. If we look at the indoor cannabis, it's 92% with 7% sesquiterpenes. And these sesquiterpenes are responsible for the unpleasant smell in cannabis. When you think about the good aroma, that comes from the monoterpenes. Most of the terpenes in cannabis are myrcene too, and I'll show that here on this next slide. Beta-myrcene is the most common terpene found in cannabis. We have fiber type for hemp and drug type, and alpha-pinene, terpinolene. These are the most common cannabinoids found in cannabis, and they're in descending order. Running a little bit out of time, so I'm going to go through this. Some of the things we do at Cannabis in the lab is we extract the hemp with supercritical CO2. We process that by decarboxylation. We can distill it. We can make vaporizer oil. We can really make a whole host of products. But one of the things I'm most proud of is the CBD Simple, and this is what we've done the Cannabis Cup with. We've recently obtained crystals. So we have CBD crystals now, and I think that's fantastic. So whenever you choose to purchase a cannabis product, whether it's hemp or anything, you want to know that the company that you're purchasing from meets certain quality control guidelines. You want to make sure that they can meet your quality expectations, your volume expectations, and some of the ways that we try to do that is by testing our product. We test it for cannabinoids, terpene levels, pesticides. We test it with microbial contamination, residual solvents, and heavy metals. So in conclusion, cannabis has a whole host of valuable nutraceutical components. These include cannabinoids, terpenes, fatty acids, and fiber. And hemp is a chemotype 3 variety with high CBD levels with very low THC content. Thank you for your attention.